Hey guys, sorry I'm not here today, but as always, even though I'm not here, doesn't mean the learning stops. So we're going to go ahead and discuss 2 learning targets and then talk about what you need to accomplish the rest of class today. Same as when we're going through material in class when I'm there. Expectation is that while you're going through this video, you are taking notes over stuff, and if you have any questions, when I'm back tomorrow, make sure you have those questions ready so you can go ahead and ask them. So we're going to start with learning target 4.6, which is about sexual versus asexual reproduction. So if you remember back to the beginning of the unit, we said there were 3 big reasons that cells would divide. Cells will divide to grow. That's how you all went from being made of only one cell to being made of millions and millions of cells. Cells will divide for repair and replacement. If you have cells that have been damaged or have just died throughout the normal life of a cell, you need to be able to replace those. And then the 3rd big reason we said was for reproduction. And that's what we're going to focus on here today. So sometimes cells are dividing to create entirely new organisms. And those organisms can be produced through either sexual or asexual reproduction. And different types of cell division can lead to sexual or asexual reproduction. So let's start by defining sexual and asexual reproduction, because there's a pretty big difference in what happens when new organisms are created by those 2 types of reproduction. So in asexual reproduction, the offspring that you make, the new individuals that are created, are going to be identical to the parents. They have the exact same DNA, and so they are exactly the same as the parents from which they were created. On the other hand, in sexual reproduction, the offspring, the children that are created, those new beings, are going to be genetically different from their parents. So you guys were all created by sexual reproduction. You have half of your DNA from mom, half of your DNA from dad, so you're not exactly like mom or dad because you're half of each of those. Organisms that are made by asexual reproduction, that can happen via 2 different types of cell division. So mitosis is one way to have asexual reproduction. Not every time that meiosis happens is it reproduction, right? Mitosis can also be for growth, and it can also be for repair. But sometimes mitosis produces entirely new organisms. Not in organisms like humans, but things like plants. Some types of plants, if you've ever had where you can take one part of a plant off, you can make a cutting of that plant and plant it somewhere else, and that plant now goes through a bunch of mitosis and grows larger, and now you have 2 plants. That would be an example of mitosis creating a brand new organism. There's another type of cell division that can also lead to asexual reproduction, and that's something called binary fission. And binary fission is really, really similar to mitosis. You start with one cell and you divide it into 2, and those 2 cells you create are identical. Binary fission is different though because it's happening in prokaryotic cells, whereas mitosis is something that can only happen in eukaryotic cells. And we never explicitly said that mitosis was in eukaryotic cells before, but if you think about it, you probably could have figured that out, because one of the first things that we said has to happen in mitosis is that you break down the nuclear envelope. Well, prokaryotes don't have a nuclear envelope, so obviously they're not going to be doing mitosis, which we just talked about. They're going to be doing a similar, but definitely different process called binary fission. But whether you're reproducing by mitosis or binary fission, the idea is that the offspring that are created from those new cells are going to be identical to the parent because they're getting their exact DNA, because that's what those accomplish. Sexual reproduction happens via a different type of cell division known as meiosis. We are going to come back and talk about meiosis later on in Unit 6. You don't even need to remember the specific name meiosis right now because we'll spend time on that later. But I just wanted to realize that that's a different type of cell division that we haven't talked about yet that can lead to some different types of cells than what we've talked about so far. That's all you guys really need to know about learning target 4.6. We're also going to go ahead and cover learning target 4.7G today. So all of learning target 4.7 is about the structure of DNA, and you guys have been doing this activity in class to explore what the structure of DNA looks like. And so all the specifics of that structure, we're going to come back and talk about together later. So learning targets 4.7A through F, we will come back and talk about later, but we're going to skip over those for now, and we're going to go straight to learning target 4.7G. And learning target 4.7G is all about how did we first discover that DNA is the important genetic material within your cells. So we all know that DNA is that important information that has to get passed from cell to cell in order for that new cell to know how to function. It has to get passed from parents to their children to pass all of those traits onto their children and make their kids have the particular features that they have. But how did we first figure out that DNA was the molecule in our cells that does that? We're going to go through experiments from 3 different scientists, some famous experiments that were done that were kind of the steps in helping us to figure out just how important DNA is, and that it is that material inside of your cells that has to be faithfully passed on to new cells in order for those new cells to have the properties that they have and be able to function. So the first experiments we're going to look at were done by a man named Frederick Griffiths. So these are Griffiths experiments. And Griffiths is going to be working with some different types of bacteria, and he injects those bacteria into mice and basically sees what happens to the mice and makes some conclusions from that. So first off here we have the strain R bacteria. And if you inject R strain bacteria into a mouse, the mouse is actually totally fine. It's not a type of bacteria that's bad or deadly in any way. It's what we call non-virulent, and so that mouse is totally fine. Now, on the other hand, he has a different strain of bacteria, what we call here strain S. It's a different type of bacteria. And if you injected that one into a mouse, so here's strain S, inject that into the mouse, and the mouse dies. So 2 different types of bacteria. One's deadly, one's not. Now, for his next experiment, he takes that S bacteria that we know is supposed to be deadly to the mouse, and he now does what we call heat kills it. Basically, he takes that bacteria and heats it up to a really high temperature. And as you guys should already know from stuff we've talked about earlier this year, if you take living things and heat them up to high temperature, that's a problem because it's going to cause their proteins to denature. And it basically then kills those bacteria. So if you take those bacteria that are normally deadly, but you kill the bacteria, well, you can inject those dead bacteria into the mouse. They're not going to have any effect. So in this situation, the mouse is totally healthy. In his last experiment, he actually mixes 2 of these different bacteria together. So he takes that R strain bacteria, and these ones are alive, but we know that a living R strain doesn't do anything to the mouse. And then he takes some of this heat-killed S strain that also didn't kill a mouse, and he mixes those 2 things together. So he's taking these 2 things that independently left the mouse healthy, injects both of them together in a mouse to see what happens. And what happens is the mouse actually dies. So even though these 2 things independently weren't able to kill the mouse, when they're mixed together, all of a sudden they affect the mouse. And even more surprisingly, if you take that dead mouse and you take a sample of his blood and you look in his blood, you can actually find some living S strain bacteria in his blood. And so what Griffiths figured out is that even though this S strain bacteria is dead, parts of that S strain bacteria are still going to be there. They may not be functioning and working in living things, but you're still going to have proteins there, and you're going to have DNA there, and you're going to have some carbohydrates that are left behind from those cells. So he figured something from these dead cells must actually get passed to these living cells and turn these living R cells into S cells. So Griffiths figures out that there's some material that can be transported from one cell to another cell, or even just from the remaining parts of dead cells to another cell, that can actually change how those cells function and what their properties are and give them brand new properties. In this case, that property making it those cells can kill that mouse. Now, Griffiths doesn't figure out that DNA is that important information that has to be transferred. We all know today that DNA is that important information in your cells that tells them how to function and gives them their properties. Griffiths didn't figure that out. But he starts to figure out that there has to be some important controlling information, and that information could be passed from cell to cell. This same type of experiment that Griffiths did, we're going to basically repeat the same experiment, or someone by the name of Avery is going to repeat the same experiment, but he's going to make one little addition that's going to answer some more questions about our genetic material. So the next experiment we're going to look at is an experiment done by Avery. This is Avery's experiment. And Avery's going to repeat what Griffiths did. He's going to take that R strain that was a non-killer, the heat-killed S strain, which now that it's dead, doesn't kill the mouse, and he's going to inject both of those into the mouse. He's going to do that. We're going to show 2 different times here. He actually did some different additional experiments as well, but we're going to look at these 2. But he made one addition to what Griffiths did. When he injected those things into the mouse, he added something else. In one situation here, he adds what's called a nuclease. And in the other situation over here, he adds what's called a protease. And a protease is something that can destroy proteins. And a nuclease is something that can destroy nucleic acids, like DNA. So he takes those things, injects them into the mouse, And here's what he finds. He finds that when you add a nuclease, the mouse is healthy. And when you add the protease, the mouse dies. And this is what helps us to realize that DNA is that important information that gets passed from cell to cell. Because what Avery figured out is that if DNA is what's being passed from those heat-killed S strains into R strain cell, if that's the important genetic information that has to be passed, then what he figures is happening is that DNA is supposed to be passed from this cell to this cell, but if I add a nuclease, that DNA gets destroyed. And if DNA gets destroyed, then there's nothing to be passed into these cells. And so now I just have my normal R strain, which were never a killer, and so they're not a killer type of cell and the mouse lives. If I had a protease, on the other hand, who cares if I destroy the proteins? What's important is that the DNA from these cells gets passed to these cells. And as long as that DNA can be passed, which that DNA is totally fine here because I only used a protease, as long as that DNA can get passed, then the DNA from those S cells gets transferred into those R cells and gives those R cells new properties. And that property that it's gotten from the S cells is that it can now kill the mouse. So Avery does this great experiment that helps us start to realize that DNA is that important information that can and needs to be passed from cells to cells and gives those cells the particular properties that they have. Now, Avery's experiment's pretty cool and straightforward, but when it was done, a lot of people still weren't really convinced. Because even though we haven't talked a ton about the structure of them yet in class, what people knew at the time is that DNA is a relatively simple molecule and proteins are a lot more complicated. And people found it hard to believe that it's the simple molecule, the DNA, that can do the super, super important stuff. There were some people that figured DNA is just too basic and simple a molecule to be this important and control all these things happening in cells. And even though the evidence wasn't supporting it, there were some people that held on to the idea and wanted to keep believing that it was actually proteins that were the important genetic information. So in addition to Avery's experiment, another experiment gets done a number of years later that reinforces Avery's findings and finally gets more people to start believing that DNA is in fact that genetic material. And those experiments were done by Hershey and Chase. So this is the Hershey and Chase experiment. And what Hershey and Chase did was they used these right here, which are viruses. Okay? And viruses are really cool for testing this question because what people are trying to figure out is it the DNA or is it the protein? Which of those is the important genetic information? Well, really simple viruses like this, some viruses are more complicated, but really simple viruses like this are only made up of two things. They're made up of DNA and they're made up of protein. So if those are the things we want to test to figure out what's the genetic information, viruses seem like a great tool we can use to test that. And what viruses like to do is they infect cells. So these viruses here are going to infect a bacterial cell, and the viruses essentially take over the bacterial cell. They start controlling that bacteria and making that bacteria do things for the virus. So again, we're going to do this experiment twice, and we're going to do slightly different things each time. So in one experiment, they took the protein, and so all this outer stuff here that's highlighted in pink, that's the protein. And the DNA is right there inside of this protein up here in what we call the head region of it. So they took just the protein and they made it radioactive. They basically labeled the protein. So the protein's labeled, but the DNA is not. And then they do a corresponding experiment where they label the DNA. So the DNA here you can see is glowing in blue, but they didn't label the protein. So in one, the protein's radioactive, and the other DNA is radioactive. You take those viruses and you let them infect the bacteria, and then you come back a while later and you look at those bacteria. And their reasoning was, if viruses are basically able to take over and start controlling the bacteria, then whatever that genetic information is, which controls cells and helps us figure out how to function, must end up inside of those bacteria. So they're trying to check to see what gets inside of those bacteria. And what they found is that in this situation where they had labeled the protein and made the protein radioactive, they don't see any radioactivity inside of their cells, meaning that protein never makes it into the bacterial cell. On the other hand, when they made the DNA radioactive, they do see some radioactive signal coming from the bacteria, meaning that DNA did get inside of the cell. And so their big conclusion then is DNA must be the genetic material. If DNA can get inside of a bacterial cell, but protein can't, and we know that those viruses are somehow taking over those bacteria, then it has to be the DNA that's inside of that cell that's basically starting to control and take over that bacteria. Therefore, DNA must be the genetic material. Okay? That's learning target 47G. So that's it for notes for today. But I want to explain to you guys what you're going to be doing for the rest of class. So today's assignment, basically you need to continue working with that model of DNA that you were building last class. So if your group never got a final model that you think is correct, if you think you still need some time to build, you need to get your picture that I told you need to take, then there's some beads that are up front on the table, and you can go ahead and grab a bag of those beads and continue working on your model and make sure once you think you have it, that you go ahead and take a picture. When you have your model completed and you've got your picture, then you're going to begin working on your write-up. And even though you built this model as a group, each member of your group is going to have to do this write-up individually. So I want to talk a little bit about what your write-up is going to look like. So basically you're going to want to open a Google Doc or a Word document, something that you can type into, and here's what you're going to create. Okay, so at the top of your paper, I want you to put your guiding question, and our guiding question for this lab is, what's the structure of DNA? And then I want you to put your claim. And your claim for this is not a statement, it's not a sentence you're writing out. The claim is going to be the picture that you took of your model of DNA. And if this is what your picture looks like, it is not correct, because this is not what DNA looks like. Because this definitely would not meet all of those criteria that we had in class. But whatever your picture is that you took, you're going to put here as your claim. And make sure somewhere with that you also include a key. I didn't type out an entire key here, but all 6 of those different colors that you had in your model, you need to state what each of those 6 beads colors corresponded to which molecule. So I need to know if sugar is the white bead versus the red bead versus the blue bead. You've got to have a key that details that. Okay, then you're going to go through and take each of those pieces of evidence you had. So remember, you had 8 pieces of evidence that helped you build your model. And you're going to take each piece of evidence that you had. And so piece of evidence 1 was that DNA is a very long molecule composed of smaller molecules called subunits. You're going to type out that piece of evidence exactly as it was on the sheet that I gave to you. And then you're going to provide a justification for that evidence. Basically, you're going to tell me, here's the model that I built, here's the evidence, and justify for me that your model and your evidence match, that the model that you created fits each of those pieces of evidence that you have. Okay? Some of the evidence may only require 1 or 2 sentences to describe how your model and that evidence fit together. Others may need a small paragraph to do it. Different pieces of evidence will be different, but each piece of evidence has to be discussed how your model meets those criteria. So piece of evidence 1, then type out the justification that explains that. Then piece of evidence 2, type out the justification that explains that. If you don't have that sheet that lists all those evidences anymore, if you go into Canvas and look on Tuesday's date on the calendar, which is when we did that activity, you'll find the link right there. So where it says DNA structure using this data, that data sheet is what you want to pull up. And so if you click on that right there, it'll take you to that data sheet that you guys had in class. And so you'll just copy, DNA is a very long molecule composed of smaller subunits. You'll take that and just paste it right in for evidence 1. Type your justification. Then you'll go back to that sheet. You can copy and paste number 2, and then you can go ahead, you can go ahead and type piece of evidence number 2. Now write out your justification. So you should have 8 different pieces of evidence and then a separate justification for each of those pieces of evidence where you tell me how this model fits with that piece of evidence. Convince me that your evidence is supported by that model that you have created. Okay? All right, so your goal then, once you've completed that document, once you've typed up each of those pieces of evidence, the justification, you're going to submit your document to Canvas, not to Turnitin.com. Most of the stuff we do, we've turned into Turnitin.com, but this one needs to be submitted to Canvas by midnight tonight. And you've got the rest of class to work on this, so you guys should be able to get this done. The reason I need it at midnight tonight is because we're going to be evaluating each other's justifications tomorrow in class. We're going to use peer editing in Canvas. And so I need everybody's papers uploaded in Canvas. I know we did this once before, and I told you that you could bring in a paper copy or you could upload it to Canvas. That's not what we're doing here. Everybody needs to get it uploaded to Canvas. So get this document done, get your homework done, get it uploaded to Canvas, because you're going to be peer editing. And part of your grade for this is not just going to be how good is your justification, it's going to be how well did you peer edit. How well can you evaluate other people's justifications? And if you don't have a document submitted, then I can't assign you papers to peer review, and you're going to miss that part of your grade and you're going to fail this assignment. So I need you to get this paper done and uploaded to Canvas. Many of you have uploaded stuff to Canvas before. If you haven't, though, here's what you're going to have to do. Log into Canvas, go to your honors bio course like you always do. You're going to start there on the home page, click on assignments, and you're going to scroll down to the one that says upcoming assignments and then the structure of DNA argument. So when you're ready to turn in your assignment, here's where you're going to go. You'll go ahead and click on that. And if you look through this, it's got some additional information on what we're going to be doing. So when you guys are peer editing and when I eventually score your stuff, here's the rubric you're going to be using. So did you provide a claim? Is your picture there? Yes or no? Is a justification for piece of evidence number 1 provided? Yes or no? And then is that justification acceptable? Does it need more detail or is it perfectly fine? Yes or no? And then you'll find them for each of those. For piece of evidence number 2, is it provided or acceptable? Same thing for number 3. If you keep scrolling, oh, sorry, not scrolling, back up at the top. Back up at the top, there's a spot that says submit assignment. And so once you have everything typed up and you've got either your Google Doc or your Word document or wherever it is that you've decided to save it, you're going to go ahead and click submit assignment. And then it'll take you right here where you can do a file upload. So choose your file, load it up there, and then submit your assignment. Okay? Please make sure that you guys get this done. Use your time wisely in class today. That way, if you try and upload it and you have problems, you've got a bunch of other classmates here. Somebody's going to help you to get that uploaded. But it's super important that you get that uploaded so that you are ready to go in class tomorrow for proofreading. Okay? Email me if you guys have specific questions, but really try and work together to help each other. Be productive the rest of class. Get it done.